Let's take a look at what you're doing in this final week of the lab course. We are in Module 5. We're going to look at statistics, summarizing research, and synthesizing research. In your Week 5 overview, you see a brief overview of what we're doing this week, in addition to the objectives and the weekly activities. In your learning materials, you have a brief description here, and then you have your weekly lecture video. For the assigned materials, under the content, you have several links on Mendelian and advanced genetics, because the lab you're going to be doing this week is on genetics. You're also learning to summarize and synthesize information and run statistics, so you have some links here on how best to summarize scientific information, and then you also have some links here on statistics. In your first assignment this week, you will be doing a discussion. In this discussion, for your initial post, you will select a poster from one of your weekly posters from 1 through 4. You will create a PDF of that one poster, and then you will also post a statement about the most important aspects of that poster. So if you were telling someone the kernel of information about your research, what would you say? The bigger part of this discussion post are the responses to your peers. You are going to respond to at least 2 peers, take a look at their poster. You're going to do some external research about that information. Several of the links here go to news websites. Those are a good place to start. When you go to these news websites, this is an article that's geared a little bit more towards the general public, but usually embedded within that article is the link to the actual scientific journal article. It's good to read the news, but if you were going to include this as a literature cited, you're really better off going and reading the full article, not just the news article. After you've done a little background research, you are going to create an oral summary of that poster. What was that poster telling you? So if you went to a poster session and you saw this poster, and then you go back to your school and to your lab mates, and you're going to tell them what you saw on that poster. You're bringing this information back to other people that haven't seen it. Each one of those should be about 2 to 3 minutes in length. There's a link here on how to do a screen capture, so you could have the PDF of the poster up while you're explaining what's going on. And then your final assignment for the week is going to be the genetic cross data. And what this is trying to do is, one, we are looking at genetics, but the other thing is we are looking at statistical analysis of data. There's some different links looking at genetics in fruit flies and some basic and advanced Mendelian genetics. There's also a link here that describes a chi-square analysis. The chi-square analysis is typically used when you're looking at data where you are counting something, and you're counting something in groups. In this case, let's say we're looking at white eyes and red eyes in the offspring of a particular genetic cross. And I've counted how many I have of white-eyed flies and how many I have of red-eyed flies. That's what I've counted. I also know, based on what I understand about Mendelian genetics, what I expected to find. Maybe I expected 3/4 of my offspring to have red eyes and 1/4 to have white eyes. So let's say I counted 100 offspring. So I expected 75 reds and 25 whites. That was my expected. What did I actually observe? And so the chi-square allows you to answer the question, are my expected and my observed close enough for whatever I based my expected on, that Mendelian pattern, to be supported, or are those numbers too different so that I need to basically reject that hypothesis? If your observed and expected are exactly the same, your chi-square is going to be 0. And there's no difference. Absolutely, this is falling under Mendelian principles. As the expected and observed values start to get further apart from one another, they're not exactly the same, that chi-square value is going to start to get bigger. And it's going to get bigger and bigger the more our observed deviates from our expected. There comes a point at which that observed and expected deviation is so great that we now reject that original hypothesis. If the chi-square value gets to be too big, whatever we based it on is not actually happening. Something else is going on. Does it seem like the observed and expected are in line, or are they too different? Now you're going to be using a simulation called FlyLab. First go here and get an orientation to see what FlyLab is all about, and then you're going to do these different crosses. Make sure you follow along with each of these steps. The FlyLab is pretty self-explanatory. Let's just take a look at this monohybrid cross. You click on the FlyLab. One of the first things it's asking you to do is to reset the traits, and then we're going to choose a wild type female and a vestigial wing male. Reset. This is our female. She's wild type, so we're going to select her. We're going to select wing size here, and we want vestigial. So we have our female who is wild type. That's what the plus typically means here. And then we have our male who is vestigial. So you can see his wings are not fully developed. And then we mate them. And so it's going to give us numbers based on that. Now that we have those offspring, we want to mate them. So we're going to create another cross from those offspring. So typically these crossings that we're doing when we're doing Mendelian genetics, we will have our parental generation, we'll create our first generation of offspring, we'll mate them with one another to create the second generation of offspring. So now we need to go in and create that second generation. So after we selected our parents, we had our female and our male. Female was wild type, that's what the plus here means. Male was vestigial, and then these are their offspring. This is wild type, so is this. So you see his wings are okay. We had a total of 488 offspring that were female wild. We had 511 that were male wild. Now what we're going to do is to cross those offspring. So we had a parental generation. We had what's known as the F1, which is their offspring. Now we're going to breed the offspring to create the F2. We're going to select this one to mate and that one to mate. Now we have the offspring of that cross. We have wild females here, wild males there, vestigial females, and vestigial males. And these are the numbers that we have of each type. Now, one of the things to notice is that we don't see a huge difference between males and females. We have almost the same number of vestigial males and females, and almost the same number of wild type males and females. We don't think that this is something that's correlated with their sex. It's not sitting on a sex chromosome. One of the things that the instructions are telling us to do when we go through and analyze this, we're going to ignore the sex of the flies when we test our hypothesis. We're going to go back to the Analyze tab, ignore the sex of the offspring. It's taking all of the wild, both males and females, and throwing them together into this number here, 742. It's taking all of the vestigial, both males and females, and adding them together here. We also want to click this box, include a test of the hypothesis. What we now want is what would we expect for the hypothesis? If we have 977 flies total, if this is a simple dominant trait, wild type wing is dominant to vestigial, Mendelian genetics tells us we should get 3/4 wild type and 1/4 vestigial, 75% wild, 25% vestigial. Now what we have to do is to look at our number here, the 977, and ask what is 75% of that. So if I take 75% of 977, it's actually 732.75. Typically we go ahead and leave these decimal places in for now, even though we know you can't get 0.75 of a fly, we still leave them in for now. And then if I take 25% of 977, I get this 244.25. So you'll have to do that calculation. What it's going to do is calculate my chi-square value. My chi-square value is right here, 0.47. It's a very low value. Remember, we said it would be 0 if these were exactly the numbers we expected. It's not 0, but it's 0.47, which is still very low. When we come in here and look at the level of significance, the p-value here is 0.4943. Remember, it has to be less than 0.05 for us to say that they deviate too much from our expected. So these values are not deviating from our expected. We accept the hypothesis that these data are in line with classical Mendelian genetics. We base this hypothesis on the fact that the wild type wing was dominant to the vestigial wing. And that's what this is showing us, because we are getting that 3-to-1 ratio that you would expect in your F2 for that type of a trait. So you'll do the same thing for the other crosses that they list in the assignment. It's also asking us to add the results to the lab notes. When we come back and look at our cross right here, we will add this to our lab notes, and you can put a comment in here. So we've done this for our first cross. You'll do this for the remaining crosses. Down here, it's asking you to design your own cross. Think about some of the other things that you've looked at here. Come up with a different cross than the ones that we've looked at. What is your justification for your hypothesis? And run that analysis. On your poster, you're going to answer each of these questions for each of the different crosses that you did. And then you're going to take all of that information and you're going to put it onto your poster. You will do this very much like you've done the previous weeks. One thing they're asking you to do that's a little bit different this week is, instead of an introduction, it's asking you to include an abstract. Read through here about what an abstract includes. If you need more tips, there's a link here. You have your grading rubric below. As always, if you have any questions, please email me and I can either answer your question in an email or we can arrange to meet virtually. I hope you have a great week.